the session number 2 of Project Pitches. We are supposed to have this time for about 9 slots before we do the 3:30 break, and we have 7 scheduled. Like yesterday, if you go on the schedule page, you have the list. You also have the link to the repos. I quickly checked. I think there's just one repo, which is this one by Vladimir Gruza, that has like no issue. So please go raise issues to Vladimir's project.
Just lay it on me, guys. This project stinks.
No, it needs a bit of love. So please give like GitHub hugs to Vladimir. And I guess we're just gonna get going.
Can you refresh?
Can I refresh? Oh my God, there's last minute changes.
Well, just one presentation.
Okay, why don't you refresh? Okay.
Just next one.
So is it for this session?
No, next one.
Okay, perfect. So I think we're good to go unless there are questions, concerns. I feel like I had something to say that I forgot. Yeah, I do actually. I was. God, where was that? Here. Yeah. Before we start, just real quick, there was a question that I was asked a couple of times. Is like, how common is it to work with this, like the tools you're presenting, all those like Python tools and things like that? And my answer was generally like, I don't know, but it's probably, my rough estimate's like 5% of people. Mathieu Boudreau, who's in Nicolas Tikov's group, just finished a little survey of papers published in MRM, which is one of the main journal in MRI physics, I would say. Is that like MRI engineering? And out of 470 papers that he found, he tried to see if they were like sharing their stuff. He found that there was about 52 papers who actually shared code or data, which would represent 10% of the published papers. That MATLAB is 80% of the languages used, which kind of, I think, fit what was going on in HBM, more or less. That's my feeling too. And that when the code is available, it's most generally hosted on GitHub. So I'd say that's the only sort of mainstream thing we're doing. And out of the paper, how many shared enough documentation to potentially try to reproduce the figures, and that was roughly at 10%. So it says really one of the core objectives of that school is to bring a new generation of scientists who actually are in that 10%. So I'd say you're kind of like fringe punk, like say it the way you want. It's not like the mainstream thing, but I think this is a very necessary change. And hopefully, the other image I've been using is like your seeds. That you're going to go back to your labs and like grow into a forest, hopefully. We'll see. But that's kind of like the hope. Anyway, after this little brief introduction, I think we should get going with the project pitches, and we start with Mathieu.
So, hi, I'm an undergrad student in computer science, mathematics, and statistics, and I study the performance of big data pipeline, and that's kind of why I came to the school, because I use mostly neuroimaging data since they're fairly large. However, one of the problems right now we have in data-intensive pipeline is that the data transfer time takes a large amount of time. And so does the transfer of a container image. So my project is going to try to reduce the container image size, and we hope that it could like decrease the time of the pipeline by a fair amount. So the dataset I'm going to use for my project is the core dataset. I probably won't use all of the subjects since it might take a lot of time to like benchmark the different pipelines. So I'm probably going to take a set of between like 100 and 400 subjects. For the tool I'm going to use for my project, I'm mostly going to use Bash to write my pipeline to reduce the container image size. I'm also going to use Docker and Singularity to containerize my pipeline. And also, since most of since I'm trying to reduce the container image, I'm mostly going to focus on Docker and then transfer them to Singularity using Docker to Singularity. And to run my benchmark, I'm going to use Dask, which is a distributed scheduler, which I'm going to set up on my cluster. For the application I plan to use, I'm going to use the BitZap example, BitZap fMRI prep if I have time, and also the Maggot Brain pipeline. So initially, there was a tool called NoRDocker that seemed promising, doing similar jobs. I wanted to benchmark it. However, I've run into some issue in the last 2 weeks, and it seems like it might not work. So I've raised like an issue on their repo and tried to explain them like what wasn't working about their framework. They also like did a custom version for Greg in the past. I tried that as well. It wasn't working. So I'm currently on hold with them. So my second idea after that was to implement my own pipeline. So I'm currently using ReproZip, which is a tool that allows to trace every single file that's being used when a pipeline is run, such that we can remove the files that aren't used, which allows the reduction of image size. I'm also thinking about implementing another script with strace, which is a similar tool as ReproZip, but it's a little bit more granular. After that, once I reduce each Docker image, I'm going to transfer them also to Singularity since it's really convenient to use an HPC. And I'm going to try to benchmark on the pipeline if it actually reduced by a fair amount the execution time. For my infrastructure, I'm going to have one node that's going to serve as a scheduler with 1 core and 7.5 gig of memory, and 10 dedicated nodes with 4 core each and 15 gig. And the bandwidth in my network is going to be 10 gigabyte. For deliverable, I want to put my script on GitHub, of course, and also want to containerize it so it's easier for people to use it. And I also want to give away to the community the minified image, since like it's used as to duplicate the work for multiple people, as well as doing a mini report of the benchmark I'm going to produce. And also, in a previous work, I did like a script to generate iterative Gantt chart that allows me to see when my pipeline execute, all different subjects are run, and it helps a lot to debug. So I also want to share that. And that's pretty much it.
Alright, so it's very technical project. Very much looking forward to see the results, because so you mentioned how big is fMRI prep Docker container?
The last time I checked, it was around 15 gig, which can be like fairly large, especially if you have a cluster and you have, let's say, like 40 nodes. You need to send the image to every node. It can become like a large amount of data.
Yeah, so that would be actually more like the data set is 400 gigs.
Yeah, exactly. So like the image itself could be bigger than the data set, and could also take longer to transfer the image than to run the pipeline, depending on which kind of pipeline you run. So that's a big issue right now.
So I'm not sure to understand that. So is that like the image, does it go from your main node to the compute node, or does the compute node re-download the image from, say, Docker Hub somewhere on the internet?
So right now, I will store the image on an NFS on like a storage node,
Okay.
which will probably be the scheduler as well, because like I can use it for 2 things. And every instance that's called a worker is gonna pull it at first, just download it from the NFS, and we hope to like cache it, because it will save a lot of time as well. So it will have to be downloaded one time for each instance.
But by download, you mean transferred from the master node, or
Exactly,
wherever your distributed
Mhm.
file system lives to the execution node.
Yeah, exactly.
And that happened at 10 gigabit per second.
Yeah, but we had some issue since the NFS only used one hard drive. So it was really slow. I think it was limited at around 500 megabytes per second. And when it's shared among multiple instances, it can like take a large amount of time.
So, okay. Because 500 megabytes is half of 1 gigabit.
It's 500 megabits. Sorry.
Okay.
Yeah, because the disk bandwidth is around 500 megabits per second.
Uh-huh.
And in my previous experiment, I did. I was using BigBrain, which was segmented in chunks of 600 megabytes, and to transfer 64 of them, it was taking around 400 seconds, which was fairly long.
So it's really the I/O, the access to
Yeah,
the...
exactly.
Who knows what an NFS is? Okay, so I think we need to make sure we give proper context. Like that's something that's really a challenge. We're in a
Mhm.
multidisciplinary environment because there's lots of jargon that has gone on here. So can you, NFS?
Quickly, an NFS is a network file system. So it's basically as if you were to open your Finder, like I don't know the name on like Windows. Only know it for Mac. Yeah, your file explorer. It will be kind of similar to that, except that it will be on another computer. So you will be limited by the bandwidth. But in my case, since I only have one hard drive, because it's not paralleled, it will be limited by the disk, since there's going to be a lot of access from the different instance towards that one disk, and it's limited at 500 megabits per second, which is fairly slow.
Yeah.
It's further.
So it's a distributed file system, like it's a disk that's shared by many computers.
Yeah, basically.
And so it's not as fast as having your own hard drive on your own computer. And actually, I was thinking it'd be great to have a little field trip to go see a supercomputer, how those things are actually wired, because it's pretty cool. It's lots of cables.
It is.
All right. And the other thing I think that you did not specify that's important is like the bit and the byte. The bit is 0 or 1, and a byte is an octet. It's 8 bit. So there's a factor 10 between a gigabit and a gigabyte.
Yeah.
So that's where we were a little bit confused at some point.
Yeah, so let's say you were to transfer like 500 megabytes on a 500 megabits network, it will take like 8 seconds.
So when you give his number, it's very important. The big B is 10 times bigger than the small b. So to understand those units, it's an important detail. All right, is there any other geeky or non-geeky feedback?
Just not a question, but a comment for people that are using the clusters. Kind of what he's talking about is of relevance to us. If you talk about working on Scratch Space, which is designed for high-speed parallel I/O versus a home directory or some other area which isn't designed for it. So if you go through the Compute Canada documentation, they give you advice on you should do IIO workloads on the Scratch directory because it's really meant for that. And if you do it on a slower directory, your jobs could take much longer.
It's relevant period because if he succeeds, you may be able to run your fMRI prep pipeline faster. For those who do fMRI prep, down the road it will benefit you. All right, so thanks a lot.
Thank you.
So, next on stage, Christophe.
Alright, so my project, my name is Christophe. I'm starting my master in September. I just finished my undergrad in psychology at McGill, studying in the IPN at McGill. And I've been working actually on some of my project for the past 3 months. So the structure itself is pretty much the slides. But what I study is actually chronic low back pain. So this idea that people actually sort of have an injury or some issues. In general, chronic pain is, but my project specifically is on low back pain, which is very common. And throughout months, although we can't really report any sort of site of injury, they still report pain. And why is that? That's sort of the whole field of it. So essentially, and it's really mixed between the sort of comorbidity factor with a lot of depression and anxiety. On top of this, there's the whole pain or sensory factors and bit of inter-individual differences, such as like interoceptive awareness. So everyone sort of, or less or more sensitive to how they feel their sensation, and this sort of whole chunks of information, which sort of trying to maybe through brain imaging to sort of segment them, or maybe find to supplement what we know from the behavioral or questionnaires. All right, so which dataset I will use? Okay. I actually use OpenPain, which is an online resource for only brain imaging on chronic low back pain, which is fairly recent. And it's mostly MRI for now. You can actually have the access to the site. It was done on a prior study on placebo, but like the actual effect of placebo as a treatment itself. In our case, what we are just going to use is those that did not receive a placebo. So they actually did just being monitored, that's it. And yes, and essentially, it was an RCT study done on placebo, and we're using the T1s and resting state fMRI. In my case, in this project, I'll just look at the resting state. And which tools do I want to learn? The first one is fMRI prep. Obviously, it's a reproducible pre-processing pipeline, as we all know it. And my goal is sort of here is just to get a sort of a draft of the scripts I will be using during my master and probably in my lab. And that was the first one, to just sort of get to know more of fMRI prep and to install it on Compute Canada, which I already started. And my case is Beluga, which is sort of the more recent clusters, and because of this, some of the stuff I feel is actually, let's just say that there's better visualization on like Cedar and other clusters, so I'm sort of getting my way around. I want to learn Python. So far I've only used R, and I'm trying to transfer to it, although, I mean, they're both great. And obviously Jupyter Notebook as well. For visualization and just the narratives of my script, and I learn as well. Also, for my resting state, I will also use, as brain population, the Scheffler 2018, which also includes the Yale is in it, so it's like the 400 population and the 7 ones, as well as the Power and Peterson, which is 264. And you can actually get the network modules from a file they actually provide. And the whole point of having two is actually just to validate whatever you get in two different ways. And the actual functional connectivity tool that we'll be using is Net Neural Tools, which is in Python, provided by the student Ross, I think, from Bratislava. And I will use the bootstrap ratios that you mentioned as a sort of a cross-validation tools that's just specified for their package. To sort of get a better sense of the actual how the brain imaging relate to pain, we'll use some questionnaires. 3 of them are mentioned there. The McGill pain questionnaires, which is really about the quality of pain, this location, and actually and the site. BDI, so Basic Depression Inventory, and also Ecological Inventory Assessment. I don't know if you've heard of them. It's just basically on your cell phone, you sort of record symptoms or question every day. You get sort of a notification, you answer it throughout 2 weeks, so you actually get a real-time sort of information in a real context. So, and for the deliverable, what I'm going to want to use, essentially have all the scripts just to install and operate fMRI prep on Compute Canada, so it's actually sort of available to other maybe members of my lab. My Python script for my functional creativity, just present my output of fMRI prep. Yes, actually transfer and make the script on a notebook with all the necessary explanations, and And yeah. And the medium that we'll use obviously is my notebook. And I will create in a repository just all the little scripts that I use for my whole project. I mean, for BrainHack. So that's it.
Alrighty, so there's a few people doing kind of like fMRI prep Nyler.
Yeah, it's a great tool, I feel.
We can be a little group, study group right there.
Mm-hmm.
Yeah, I see Anthony yesterday.
Mm-hmm. Yeah, I'm actually sitting beside him and we're doing everything together.
Okay. So perfect. Hopefully you can work hand in hand and help each other.
Mm-hmm.
Are there some questions here?
Yes?
Very random question. What are you using as the software for EMA, the ecological materials?
The EMA software that is used was on the actual OpenPain dataset. I don't know because this study was done like 2 or 3 years ago in Chicago. But we're gonna start our own, and I don't know because there's actually some new ones from I think ACO. Some of the researchers relate to ACO. I mean, in psychology, just got a new one. I don't know yet, but I mean, it's a good question. If you have one to recommend, I'm open.
I've worked with Metric Wire.
Okay.
I can show you a little bit how it works.
Oh yeah, that would be nice. Yep.
Maybe like data set.
Mm-hmm.
Is that the data set for your master's project?
First, yeah, part of my master project. Yeah.
So you're saying it doesn't have structural imaging, or are you planning to actually run fMRI prep on?
Sorry, what I meant is actually, so what happened is that they put in the image, and the student who was supposed to upload it, after doing face de-identification, what happened is that there was sort of a problem in few of the sessions. So like, there was like 4 sessions of scanning, and 3, 4, 5 had like patches. I guess what they tried to use and was sort of a problem with it. So when you learn about this, decided to like remove all of them. Although all the sessions, the first session was actually good, and back then I had the data, so I'm actually just using session 2. But for now, if you are to try to access the data, you won't see any T1.
So you have the T1.
Yeah, I mean, yeah. But I mean, it'll be in September. It's supposed. I mean, when the guy comes back from vacation, essentially.
But you have a full data set to work with.
Yeah. That's it.
All right. I think, I mean, it's kind of a general comment is that in terms of the media, so far it's not very, like, there's not a lot of creativity in there. Like, everybody's like, well, I have my code, I'll make a notebook, and that's it, moving on. Hopefully, maybe we can get a little bit more funk next week, Nicolas, in terms of the communication.
Okay. I mean, after all, because I've never personally done a notebook, so I mean, the next logical step is actually to do one.
Yeah.
But if there are other tools, I'm open
Yeah,
to.
so because the notebooks are going to sit on GitHub, and that's already, you know, they're kind of discoverable, and if you click on them, they're rendered like a PDF would.
Okay.
So it is actually a pretty okay way to disseminate the thing. Now there's a few, I think we mentioned Binder with Elizabeth.
Yeah.
That would be another next logical step. And I think you're going to talk about more like advanced communication tools.
Yeah, that's great. Yeah.
Just a preview, just think about, you know, how is this different? How is this unique? You know, how are you doing something that nobody else has done? And, you know, we can talk more about that next week, but I don't want to put you on the spot right now.
No, it's just, I mean, for me, it's just logical step. It's not about doing a project unique, it's just properly reporting everything I do so that it's actually reproducible. That's my main goal. I think in that sort of context, I think uniqueness comes second for me in my project. But it is true that figures, and I know figures and schematics is really important. This is what sort of grasps the attention of the reader. But for now, I'll start with the notebook.
You can always do something that's, you know, eye-catching,
Yeah? Okay.
even with a pretty standard pipeline. So,
Okay,
you know,
okay.
we'll talk about that.
Yeah, that's nice.
And it was by no way a criticism, it was more a plug for next week where we're going to have a lot of emphasis on
Well, that's good. That's
communication
creating
and visualization.
interest and momentum.
All right, thanks a lot.
Okay, so here we go. Next project. Is it you? Because I was looking by the name and it was like Jackie Chan. Is that
Yes, you'll see why later.
So hi everyone, my name is Rebecca. I'm a PhD student at UQAM studying clinical neuropsychology. And basically my PhD project, just to give you a little rundown on it, it's involving PET imaging, specifically cholinergic PET imaging, on people who have this disease or this disorder called REM sleep behavior disorder, which is actually a prodromal stage of Parkinson's disease and Lewy body dementia. So my lab's particular focus is on this PET radio tracer, which is new and which we're trying to get out there, trying to get a lot of publications out to show that this is a viable way to measure acetylcholine. So far my lab has been using statistical parametric mapping, of which there is a picture right here taken from a random publication just to show kind of what we're doing. But I know that there are other possibilities out there. And that's what I'm looking into. I'm trying to see, okay, what can we do that can maybe step this up a notch to do some other exciting things. So at a bird's eye view, I'm at BrainHack for two main reasons. The first is I need to get a handle on what my current lab's pipeline is. Right now, it's very confusing to me. And it's been challenging given the training that I've gotten so far. Like, I'm having some trouble understanding exactly how it works. So that's my first goal. And then my second goal is to learn some cool software to implement in my lab. Okay, so let's get into the specifics. Okay, so project one is to learn my current pre-processing pipeline. So I'm less excited about this. I didn't come to BrainHack because I love pre-processing. I like it because I like BrainHack because of data analysis, not because of pre-processing. I'm having trouble with this part because Mink Tools is not very user-friendly, as several of you know. And also the instructions that have been passed down to me are kind of rough. You know, I think it was just a busy PhD student who gave this to me, giving me, okay, here are all the steps to do PET imaging. You average, you blur, and it's like, okay, what files? Because I have to get this off the server of MNI and blah, 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 and it's just, I'm very confused. So this is where Jackie Chan comes in, and this is how I've felt for the last couple of days. So luckily I have a lot of people who have been helping me understand this pipeline. So that's my goal is to successfully reproduce it on Mink Tools, using Mink Tools, using Civit through CBrain, so just getting really familiar with these platforms. And then if time allows, I would also really like to run this data through ANTS, which I hear is difficult, as well as Dartel, just to see what other platforms are there aside from Civit, just to get some ideas about how maybe my lab can branch out if we choose to. So my deliverable number one will be kind of a culmination of this work in that I will have a Jupyter notebook that has hopefully better instructions than what this was. No criticism to whoever passed it down, but I'm planning to just actually break it down and explain why we do it and how, like, all the bash scripts that follow and whatnot. And hopefully it'll ease the stress of future lab members. Okay, so then project number two, the one I'm honestly more excited about, I'm going to be looking at structural MRI data. So I was planning on downloading a subset of the Prevent AD, which Alexa is working on, or Oasis, but actually Josh has offered to create some simulations of data, which might just sort of help me jump to the next step. And so my goals here are pretty simple. They follow a pretty simple path. You know, I'll be doing some feature extraction where I hope to just look at some basic morphology. I'm not looking to do resting state type complexity here. So just cortical thickness, brain volume, use NumPy, pandas, dimensionality reduction using PCA, and then maybe try random forest, hence the picture. And use NiLearn to implement this, use some cross-validation techniques that we're going to be taught next week, which I look forward to. And if I become a superstar and somehow manage to do all of this, my dream would be to actually to look at longitudinal models, so ones that can actually predict disease conversion. So I know that for this REM sleep behavior disorder cohort, we're looking to see like how long it takes before they eventually convert to Parkinson's disease. So I'd be interested in modeling this kind of thing, seeing if you can predict how many exact years they're going to develop the disorder. So yeah, so my second deliverable is basically a Jupyter notebook that thoroughly walks through these steps and with plots saved in line. And I hope to, in addition to putting all of these on GitHub through these Jupyter notebooks, I also would like to deliver these to my lab in a lab presentation. So I'm making that kind of my medium. Comments, questions? I would love any feedback. Am I overcharging myself with work?
I think you're doing fine. Like, that was great. I think people are quiet. I think 2 projects is a lot, and your first project you may sort of maybe underestimate what that represents.
Probably.
So just because one of the choices that have driven us to teach the stack, like the set of tools we've selected, because they play nice together and they're fairly easy to package and to program. And CBrain, for example, is a completely different animal. It's a web-based tool. I believe there's an API, which means a way to code what you want to do on CBrain.
Okay.
I think the closest thing we have to an API CBrain was Clouder. It's developed by Greg, but it's not CBrain.
Okay. What's it called again? Sorry?
Clouder.
Clouder.
Like
okay.
the thing you give to cats, I think. The logo is a cat.
Okay.
Or chowder is what you
Chowder?
And so that's right. Whatever. It's got a cute cat logo.
Okay,
Greg,
I'll look for the cute cat
Greg
then.
makes great logo. But I think you can spend your BrainHack school on this. Like using, and those are not very mature tools necessarily.
Okay.
Like we would have to discuss with Greg, but like some of the tools we reviewed, like NumPy, scikit-learn, have hundreds of developers, extensive testing and things like that. Clouder is Greg's master's project. Those are like very different sort of like level of polishing of the project.
Okay.
So that would be a comment that maybe you may want to focus on one thing there.
Yeah, I kind of was hoping to wrap the project 1 up really quickly because it's the one I'm less really interested in. But I need to learn how to do it because I'm going to start doing scans soon, and I need to tackle this Mink Tools learning curve.
Trust me, I've been 15 years using that.
Yeah.
I know what you're talking about. And for project 2, a suggestion would be you've got a data set that's available on ADHD 200.
Mm-hmm.
It was a competition organized by Mike Milam, the guy in New York that Claire referred a lot about.
Okay.
He was a postdoc supervisor, and he coordinated it. There's about 1,000 subjects, and it was mostly an fMRI initiative, but also structural scan. And Carlton Chu, who was at the time at the SPM place in London with John Ashburner, processed everything through Dartel and shared that before he walked into the sunset and Google DeepMind hired him. But so he's put all of that online, and you can download them. So you have all the gray matter density volume map for these 1,000 subjects.
Already grabbed from the
and
files.
all like the behavioral variables are there, including the diagnosis of ADHD. So you can just grab that and get going. Maybe select a subset of subjects, something like that.
Yeah.
And regarding the visualizations, not sure how much has been mentioned, but have you talked about plotting at all?
No, we are letting you do that.
All right, good. Because a lot of these things, it's relatively easy to make these figures interactive, make them pop up. So, it's something that would be very interesting to do, and a lot of you could recycle some of these tools, given that a lot of you are working on MRI analysis and visualization.
Cool.
Okay.
Great. Thanks.
No further comments. Thanks a lot. Very glad. I'm just wondering, just for my own curiosity, what image is WTF exactly? Did you use it?
Too many questions.
That was the first version for the Ming tools you had. I'm not recording, I won't. Just like not gonna. Hello. Anyway, moving on. Next project up is, God, I don't know, Soraya. All right, stage is yours.
Cool. Okay, well, hi everyone. My name is Soraya. I just finished my first year master in the IPN at McGill, so now I'm going to start my second year. And my thesis is broadly about memory, specifically the process of memory consolidation in Parkinson's disease patients. And so for the past year, I've been exclusively working on behavioral data. And just recently, we've collected sleep data. So it's the first time I deal with EEGs and sleep. And so now I have this data set that I need to do stuff with, but I'm not really sure what to do because no one in my lab knows how to, no one in my lab does sleep. So yeah, exactly. So I'm in this to try to learn how to, I guess, pre-process the data. And then, so we have a couple of softwares, but I mean, for the sake of BrainHack School, I wanted to be able to sort of do it like writing scripts and do my actual analysis myself without having to like use a software of which I'm not sure what exactly is happening. So the data I have is I have polysomnography recordings of about 20 Parkinson's disease patients. So these are long files because they're nights of sleep, so they're like 8 hours long. I have EEG channels, muscle activity, EMGs, and I have ocular activities with EOG, and I have electrocardiography. I have the audio of them snoring. I have videos of them sleeping. I have pulse oximetry, which is measuring blood oxygen. And so I have these set of data. And what I'm trying to do now for this, I guess, BrainHack School is I told myself, okay, well, I'll first learn how to pre-process this data from just like writing scripts myself and sort of understand also what is happening and like how. So also, by the way, if anyone works with sleep, so I've seen a lot of people have some EEG experiments, but if anyone is also familiar with sleep, let me know so then we can talk, because I am not sure if a lot of the tutorials and documentation is specifically on EEG data, not necessarily sleep, and so there are different sort of channels that you look at in sleep, so I'm not sure if I could apply everything I read from the tutorials to my specific experiment. But anyhow, so my goal is ultimately to create this figure. And so this is a very densely packed figure, and by being able to recreate this, I also need to compute all the variables that are inside this by running all my scripts before. So I need to, for instance, detect sleep spindles and slow oscillations across nights of sleep. So I could write my own functions, or I could, there's also, I think, on VisBrain, there's a way to detect these specific have a specific duration, specific frequency, and they're like sleep events. So there's a way to detect them. So I would need to detect them for each participant, and then sort of count them. Look also at generally the power spectral density of frequencies. So here this graph has on the x-axis every hour from the first hour until the 8th hour of sleep. On the y-axis is the frequency. The heat map is the power of each of these frequencies. And then above it, there's like the hypnogram, which is the specific sleep stages for every participant. And then on the y, I don't know if you, do you see my mouse? Okay. On the y-axis is another measure, which is these sort of sleep events, like sleep spindles and slow wave oscillations. And then there's like another added graph with, this is just the count of these events across, again, time. So from the first hour to the last hour, and again for the other variable, which is sleep oscillations. So I would like to be able to create something like this because it sort of forced me to learn a bunch of things to be able to reproduce that. And there's also, I would like to do sort of like topo maps, so maps of the power of, so here it's sleep spindles, also slow oscillations for every participant, and sort of where they mostly occur across the EEG map. I don't know if I have enough electrodes to do this, but it's something I want to attempt. And so ultimately, a lot of my output is going to be visual. So I want to recreate this figure, for instance, which will sort of force me to compute all these other variables. I also want to do sort of maybe an interactive graph using Plotly. Because these are very long recordings, or 8 hours, I think it would be really cool if there's a way to sort of just sort of like select variables of interest and see how they progress throughout time and sort of select these things. I think this would be really cool. And again, as I said, because I'm the only one in my lab that has been thrown into the world of sleep, I would like to be able to do a binder of Jupyter notebooks with the pre-processing of my data, but also the visualization. And for the pre-processing, I want it to be more of a tutorial so that people in my lab sort of understand the steps and what I'm doing, and sort of we could all sort of understand what is happening. But then I don't know if I could do this because I don't think I can share my data openly. So maybe it would have to be something on a private repo, but then I don't know if I can use a binder with a private repo.
So binder, private repo are not the same.
Right, I know, but like it's if I have a private repo of all my Jupyter notebooks and I want to like just pack everything so I could have all the requirements of all the sort of the packages that I use type of thing, could I share that?
On GitHub in a private repo.
Okay, okay.
That is shared only with your lab.
Yeah, exactly.
Now, something that may be better is to actually do your tutorial with open data.
Right, so that's the thing. I was looking for open data for PSG, polysomnography, and I couldn't really find.
So you may want to check the 100 Nights project. It's Nathalie Regard, she's an artist, and she's recorded sleep for 100 nights consecutively.
Oh, okay.
With a 256 EEG channels.
Okay, okay.
Set, which is
that's a lot. I have I have 20.
All of her data are open. The EEG as well as transcript from her dreams.
Okay.
And of her days.
Okay.
For the 100
oh,
days.
okay. Yeah.
So it's all like online. You Google it, you can download it.
Okay, cool.
If the format is compatible with what you do, then you've got open data, and also it's an incredible data set. So actually, it would be a lot of fun to just look at that, maybe rather than yours.
It would be but honestly, because like also I'm looking at patient data, so it's like it's not clean. So if I want to start doing something on sleep, it's better if I use sort of like. I mean, she is young and healthy, I assume. So it's like sort of like the perfect way to start in a sense.
Elsie, she's Nathalie Regard, I would say, but she's a next level artist who does crazy things.
Okay.
Like sleep for 100 days with a full EEG headset. So the headset was set up by the team of Guillaume Dumas in Paris, who's a specialist in EEG.
Okay.
So it's high quality data, I believe.
Okay. So yeah, I think I may just maybe shift to that.
She had gel on her head.
Okay,
night, so it's really like,
it's a lot of commitment.
Another thing that, again, is a way of getting around this data privacy issue is creating a synthetic data set.
Like
and again, it might be overkill for what you're trying to do, but just be aware that whenever there's a privacy issue with sharing data, it's possible to create a data set with very similar statistics that will mimic, for all practical purposes, the original data set, but it will hide any identification.
Right. Okay.
And that's something that Pierre has done in a notebook. Not sure if you showed it to them.
I have not. Now, the methods to generate the data will vary, obviously, depending on the data. It was something very different from that. But the general idea is there, absolutely. There's actually recently a review was published.
Yeah, it just came out. So maybe something I'll mention next week then.
Okay, so I'll probably look into the open data set, honestly. Yeah.
And, I mean, definitely, I think, I'm not aware, you may want to research it, but it sounds like a tutorial that may not be out there already.
Okay.
It's a small enough community that it's possible that nobody has done this kind of tutorial yet.
Okay.
In which case, releasing it would have a huge value, besides just your lab.
Right, yeah, because I was looking for stuff and I don't know. But also I may not be good at looking for stuff, so who knows. But yeah. And the tools I'll be using, I guess, M&E, VisBrain, Plotly, and BiOSP, because I also wanted to look at additional signal later on, but I think that may be overkill, so I'll just focus on this for now.
And in terms of looking, that's where we post that on Eurostars and post that on the Mattermost BrainHack.
Like just asking.
Explain what you're doing and say like if anybody knows of a similar resource or would be interested in maybe giving me feedback.
Okay.
Because I know, like people like Guillaume Dumas, for example, would be interested by it.
Oh, okay.
Possibly. Or have this, really.
Yes.
What kind of memory are you studying for consolidation? Is that
It
specific?
It's so for this experiment it was declarative. We did a word pair association the night before and the night after. But the past year I was just looking at motor learning. Yeah. So, but for the motor learning it was just behavioral, looking at across days, enhancements, things like that. Yeah.
You have experience a little bit with digits. Yes. Maybe
Inside the mRNA, and it's not
it's something different. Your signals are
night and it's a bit different in healthy adults. Yeah. But I guess Karim and maybe in the Omega database there should be a lot of make data set of sleep because I think they provide a lot of sleep in here, usually carrier. So I don't know if it's pushed into the Omega database, but
Omega?
so
maybe
not open database. It seems
Right.
you need to register, and I don't know if you can share it once you have the data.
like the advantage of Nathalie Rogar's project is it's her own data and she's putting on the web,
Right.
so I don't
Right, 'cause
think
it's
there's really like
the
any
privacy is-
issue.
yeah.
And the license she's put on it is really a Creative Commons. You can do
Okay.
pretty much what you want with it.
But if you want to find collaboration with DMLI, I think the group of Julien Voyon is doing some recordings.
Right. Yeah.
So,
He's my committee. He's on my
Okay.
committee.
Okay. All right, thanks a lot.
Thank you.
So I think we're doing okay with time. It's 2:50, so we should, be done by 3:20, 3:30. So congratulations everybody to be right on time. And now Vlad. So Vlad is the one who needs the issues. So please, again, go ahead. And if you have ideas and you're too shy, type away on GitHub.
Okay, so my name's Vlad. I'm starting a PhD in the fall in the program QLS, which means that I have actually no idea what I'm doing because it's rotations and I only have my first rotation picked. But for the first rotation, I'm working with Dr. David Rodko at the MNI, and he's an imaging physicist in MR, and he has the quantitative microstructure imaging lab. So basically what he's looking at is just, applying various MR metrics to go and look at how subvoxel microstructure varies in normal and disease states, and kind of just like develop additional tools to allow us to just a typical imaging workflow, look at it better and smaller essentially. So for the project that he gave me tentatively, I'm just going to be looking at a mouse model of demyelination. So basically you just get mice, you feed them some cuprizone for a certain amount of time, it's toxically demyelinating, and then you see how this changes, and this is basically like a surrogate for MS from what I understand. So for my data set, I'm using, right now I have data from one animal, and each data set includes like basically a static field, sorry, not a static field, a beam on image, like basically with no diffusion, diffusion weighting applied, and then it includes at least 2 additional field strengths, so that you can then use a gradient table, which includes these field strengths and the directions, to estimate a diffusion tensor. And what's a diffusion tensor? I learned this basically like this week, but a diffusion tensor basically estimates how water, basically how water molecules move along structured, axons along the brain. So basically, they're not going to move across barriers, but they're going to move along barriers. And so you can essentially estimate tractography using the diffusion tensor. So I'm working with one animal for now, but next week he's going to give me a whole data set, including animals that have no cuprizone treatment, and then 2 time points of cuprizone treatment for 3 and 5 weeks to see how myelin is changed in that time. So far what I've got done is just basically some pre-processing in Python. Because before this I was a MATLAB guy, and apparently that's not in vogue anymore, so I'm trying to do this all in Python, even though it'd probably be way easier to do in MATLAB than what I know. So basically the first thing you obviously have to do is reconstruct your MR images from raw data, but that's already done for me. And then import the nifty files, and then you have to go and essentially concatenate all these images together so you can then estimate your diffusion tensor. But before that, you gotta segment the brain out of the background using a mask, and then you have to apply a Gaussian filter to like basically suppress noise and get rid of really sharp discontinuities in the data, because when you're modeling, let's say for example the kurtosis tensor, which is like the diffusion tensor but a little bit expanded, it doesn't like that, because then you can have like singularities and whatnot. So apparently that's a necessary step. So I got that pretty much done in Python, and I'm using DiPy and nibabel to basically do all this. So it's pretty sweet because the most recent iteration of DiPy actually came out like on August 8th or something like that. So it was like super convenient where they just made this tool and then I started BrainHack and then I'm using it now. So that was pretty neat. So yeah, I'm using that to fit the diffusion kurtosis tensors, which is this figure, which I'll get to in a second. And that's done. And then I also have to look into this thing called a naughty model. I thought that's a great name for a model, by the way. But I haven't really gotten there yet, so I don't really know what it is. But I mean, you can read the words. It sounds fancy, and I'll just do that next week, presumably. So what I have done so far, like I said, is I got all these interesting diffusion parameter maps. So we have like fractional anisotropy. So how much, basically, from what I understand, it is kind of like a metric of the ordered movement of water along tracts. So like how much of it is anisotropic and how much is non-anisotropic. Mean diffusivity and then axon radial diffusivity. So I'm still kind of trying to figure out what these actually all mean and how to interpret them. And I'm still trying to figure out kind of sort of, oh, where's the rest of my thing? Did it not get refreshed?
Yeah.
Oh, that's weird.
Maybe refresh the page.
Oh, I pressed save, okay.
So yeah, no, there's a trap. Like what you see is not what you get.
Okay. Oh, there we go. It's all
okay,
good.
Great.
So anyway, so this is what I have so far. But what I don't really understand thus far is what the actual numerical values associated with these things are, because like why this one doesn't look exactly like this one, and what the relative value of the background being 0 versus what is actually occupying these voxels, like what these numerical values actually mean and if they have any physical significance in and of themselves. Because then if I'm to try to compare between conditions and between animals and whatnot, and I don't really know what these non-normalized values mean, I need to figure that out. But I mean, so far, this week's been pretty productive because, like, as of Tuesday, I didn't even know how to import a Python data set, so I'm pretty stoked on that. And then moving forward, I gotta basically, like I said, compare, generate these for each animal as well as the naughty model, and then pick ROIs. This isn't my figure, this is a figure from a paper, but pick ROIs by co-registering all these things to a Mink atlas, which sounds like it might be kind of scary and might work really nicely or might not work at all. So that's for next week though. And so yeah, I'm gonna do that across all the animals and then see if I can like learn anything interesting about how the microstructure of these axons changes in response to basically poisoning some mice. In a bonus, what I'd like to do once I actually start the project for realsies in September, what I actually probably what I want to do is take a step back from using these packages, because you'll type in like one line of code and then this thing does a whole bunch of math, like manipulating tensors and whatnot, then it just gives you a result, just like an object with some fields that you can manipulate, and you're like, okay, cool, thanks Python, that's really nice, but I really want to know what's going on under the hood. So I'll probably try to go and, let's say, if it's not terribly complicated, try to implement diffusion tensor fitting in C, for example, so that I really actually know what's happening there, instead of just kind of taking advantage of DiPy and taking it for granted. And then it'd be pretty nice to also just latex this work all up and draw some nice figures, because I'm also terrible at drawing and I'm eventually have to learn how to do that as well. So yeah, that's what I've been doing so far, and thanks for my TED Talk. Cool. Questions, comments, concerns?
So this is something that we can definitely help with. Next week you'll be around.
Yeah, big time. I saw that you guys are going to be around next week, so I'm going to be really picking your brain a lot.
So Matteo is also around. He's there. So somebody that you should talk to.
Okay.
In particular, how to interpret all this, and believe me, the math here is not very difficult.
Yeah, I saw the actual basically like the tensor exponent
So
fitting.
I don't think there's any need to implement this in C++. It's trivial.
Okay.
You can use what NumPy uses.
Okay.
And still, we can think a little bit about what to do about the naughty model, the interpretation of the data, and also, so you work with nifty files, is that correct?
Yeah.
Have you thought about converting it to a bits format?
I did, and I asked David, I asked my boss, and he's kind of just like, it's not really necessary because you don't have that many actual animals. And then I was also talking to Greg, and he's like, well, look, I mean, you could for the sake of the exercise, but unless you have thousands of participants, you don't really need to do it. And then as far as just moving this data around in Python, Nibable seems to be doing a pretty good job. So I did consider it, but I don't really see it as necessary right now.
So Nibable and beads have nothing to do with one another. Beads is about organizing multiple files. Nibable is about accessing one file.
Oh, okay.
So the library to access beads collections is PyBit.
Okay.
And in terms of sharing, I think it has very quickly some benefits, because the truth is that this is the documented format that you don't need to explain.
Yeah.
And so if you organize data, anybody who come after you after this response rotation will benefit directly from it without you having to write documentation.
Right on. I'm totally open to doing that just for the sake of the exercise too. But I'm not really sure if David's trying to share this data online either. But internally.
I'm going to see him the week after.
Yeah.
But we can touch base briefly because we collaborate, and a lot of this is very interesting to our lab as well.
Okay.
To Matteo in particular.
Right on.
So we'll come up with something that is mutually beneficial, and I think BIDS is a good idea, especially as a long-term vision. 20 animals is quite a lot actually, so it would help. But we can discuss in person, and it's an exciting project, and I guess pretty different from what everybody else is doing. I guess there's not much microstructural imaging.
There's lots of different stuff. You weren't there yesterday, but we have Marcel, who I don't know where he is, but he's studying pupil dilation when you pilot drones. So sorry,
I missed those yesterday.
Lots of stuff going on here.
I'll catch up.
And maybe in line with what you were saying about maybe it's not necessary to do it in C. One of the advantages of Python is that it's really compact and readable. And so I think there's huge values in re-implementing things, but maybe a preliminary step would be actually have a look at the code.
Okay.
Because unlike a C code, it should be compact and easy to follow. DiPy has a quite specific stance on this, that it's also using something called Cython, which is a Python with more data typings that you can compile, so you get close to C performance.
Right.
So you have things like nonlinear registration implemented in DiPy.
Yeah.
And which usually you never get enough performance of a language like Python to do those type of really intensive operations, but it is, I believe, in Cython, so it's relatively fast, but it's still quite compact and readable.
Okay.
So I think as an exercise, it's excellent to have a look at it for the science and for the software engineering.
Yeah.
Because the way they build their stuff is really modular, compact, well tested.
Yeah.
So plunging in those kind of code base is actually an amazing learning experience as a software developer.
For sure.
Which we're not necessarily software developers per se, but we build software, so we're kind of software developers.
Sort of. I mean, I bet a software developer would probably be like, no, you're not. But between ourselves, yes.
I mean, we are de facto scientific software developers.
Right.
Just the way it is. I mean, most of us, I think, here are going to code something to process scientific data.
Yeah.
Some kind of scientific
developers.
A question? No question. Dang.
Cool. Thanks so much.
So thanks a lot.
Yep.
Okay, so we're almost reaching the end, and we're cranking up the style in terms of the scientific illustration here. Matteo, thanks.
Hi, so I'm a postdoc at Polytechnique, and my project is about building a meta pipeline. Now this has two goals in mind. The first one is to actually build a system that allows anyone to reproduce any diffusion pipeline that you would read in an article or you will see in one of the many tractography challenges. And the other goal is to participate in the Iron Track Challenge that has been organized by the Martinos Center in Boston. And I'm really against reinventing the wheel, so this is actually not writing any new tool specific to diffusion, but actually to build a system based on NiPipe that would allow you to actually combine existing workflows or new ones that you can build and try different combination of parameters or tracking algorithms. And this is the very bare bones skeleton of the tool that I would like to build. In each of these boxes you would have a specific NiPipe workflow that would take care, for example, in the first block of the preprocessing and the modeling of the diffusion signal. Then you would have blocks to actually do the tracking and reconstruct the tracks. And finally you would have a block to put together all the tracks you have built and filter them based on some priors that may fit for your data. And I'm planning to do this using mainly NiPipe and Giraffe Tools as an interface. If you don't know Giraffe Tools, please check it out. It's a project by Tim van Morik from Belgium that makes very easy to build a NiPipe workflow. And I'm also thinking about embedding all this system in Boutique, so it would be easier to run literally everywhere if you have access to a cluster or to the cloud. And in terms of deliverables, apart from the actual toolbox that I'm planning to build, I would like to build a Jupyter notebook to actually compare different tractography pipelines, and I see that this can become, for example, a NeuroLibre contribution. And just a bit more of a background why I'm doing this, because usually if you look at the results of many of the recent tractography competitions, you can actually see that in most time you can identify a phase where you are just doing the preprocessing, then a phase where you are doing the actual tracking, and finally you are doing some filtering. All of them are made like this, but most of the time everyone is re-implementing all of these over and over again. And there are some examples like Tractoflow where people actually use the modern workflow systems to put together several different tools, but the thing is that they are not thought to be modular and to be sort of plug and play. So this is my project, and I would like to know your opinions and any suggestions. Thanks.
Very ambitious project. So you mentioned Tractoflow. So
yes.
I'm not closely following that, but I know there's a few pipeline. Like I think there's even like a DMRI prep project going on. How is that positioned compared to those other efforts?
So ideally you could implement Tractoflow in this system, so you just subdivide the various parts of the workflow in preprocessing, tracking and filtering. And the advantage would be then that if you want to change something, for example, you want to change some parameter in the tracking, you can do it more easily than in the actual Tractoflow. Same thing for DMRI prep.
And so to better understand your long-term goal, like, do you see that as a sort of an exercise, as a tool you want to use to enter the Iron Challenge, or as a tool you want to take over the world and become the committee standard?
I would say the second and the third. The third maybe in a little of a smaller word, in the sense that I would like to have a tool that would allow anyone who wants to reproduce a pipeline that you read on a paper and then make other people reuse that in an easy way.
Wonderful. So this is the start of something big. we are witnessing the birth.
We start from small and we see where it goes.
Did you have a chance to speak with Greg here about this?
No, I tagged him in an issue, and I've seen that he replied after lunch, but I didn't read the reply yet.
Because again, we don't want to reinvent. So
No.
Greg might probably have already done something, or if not, we'll have some good input.
Yeah.
So, Greg has this like clouder system, and he's using it to process diffusion MRI data. And I'm aware of several, initiatives. The lab of Max Decoto has pipeline. I think they just released it as a preprint. So maybe it would be worth having a community plan.
Yeah.
How do you want to approach sort of like leading this project and engaging with the community? So next week, Elisabeth Dupré, she's going to give us a short training from the Modila Foundation. I think she's calling it like working openly, leading openly, something like that. WOLO. And so maybe you can get ideas from that.
Yeah.
But I think that's the only project that has such a wide scope, but it's super exciting. And I mean, for having started and failed several ambitious projects of that kind myself, I can tell you that community aspect is critical, and you should probably think about it from the ground up.
Okay.
So that would be my feedback. All right. Thanks a lot.
Thank you.
Okay, so the last project pitch for this session, and we have still 20 minutes on the schedule, so we should be very comfortable. You can take your time.
Yes, of course.
François Lespinasse.
Yes, and this is going to do some publicity for your project, Amphia. Excellent. So I'm an undergrad. I think me and François here are the only undergrads here. I'm currently finishing this is my last 3 credits, right? And I'm transitioning to a master's project with Pierre, and I'm charged with handling physiological signals that are being recorded in the fMRI. So, are you guys hearing me well? I don't feel like I'm...
No, because the acoustics actually good.
Yeah, it is?
So, yeah.
All right. I won't play with that. I thought I had...
So the microphone, first of all, is this
it's this one. Okay.
That's for YouTube. That's for
All
YouTube.
right.
You just messed with them. That's good. That's for the people. And yeah,
Cool.
It's surprising, but it does work.
Excellent. So, okay. So my plan is to maybe just do a little part of this project that I will continue during my master's, and eventually this repo that I'm building will be merged into the repo that I have in this private GitHub repo that we have. And so the problem that I'm facing here, and I would very appreciate help with that, is canceling noise picked up by the ECG electrodes in the fMRI scanner. So as you can see down there, this is let me refresh that here. So this is an ideal model of a heartbeat, and this is what we get during the scanner. This is a 5-second segment, and so there are supposed to be approximately 5 heartbeats there, and this looks like there are heartbeats over there, but these are really gradient artifacts. So there it is. And so the package that I'm working with currently and doing some testings, it's been now 10 months that I'm reviewing the literature and, you know, reviewing what's the best technique to cancel this noise. And I've come across 2 packages that are really interesting and that make use of machine learning packages as well to verify the arch, the peak detection of the algorithm. So this is NeuroKit and biosignal processing in Python. And these are tools that I would like to, you know, study more and more. And so there's a specific filter that I would like to design, and it's based on higher order statistics that takes the distribution of data points between 2 adjacent artifacts, and considering that there's a lot of variation in that time window, the kurtosis of this distribution is very elevated. So I come across in the literature this 4th order central moment filter that normalizes this distribution and clean the signal. So this is a flow chart of the pre-processing pipeline I would like to build. Yeah, maybe just go back. Do you guys see well? I don't. So in the top here, this is the acknowledge file. So we have to, so I have to export it in the Panda object here and make it readable to Python. Then afterwards, I would like to parse the data in different blocks of the experiment, because the recordings are continuous, even though the fMRI, has different segments, different epochs of the experiment. Let's say a 10-minute segment of movie watching or video game playing. And so after I parse the data, I can take some clean ECG and extract a template from that that could be used to verify the detection of fMRI filtered ECG signals. So I would then send segments that are polluted with fMRI noise, and I have all these functions in NeuroKit that are already built, and my plan in this BrainHack is just to write a Jupyter notebook with all the functions I know how to use and that I know will stay there, and just add this filter design that I would develop during the 2 next weeks. And so these are the deliverables I think is realistic to have for the next few weeks. And so yes, I have this detailed markdown in my script here. You can just visit it and let me know what you think, and maybe give me some insights on how to parse the data in the more efficient way, and maybe help me with this filter design that I would do. That would be it. And I would like to plunge in this Twitter world and maybe do a little post and get some feedback from the scientific community up there. That would be it. Is anyone? Yeah, Vlad?
So, okay, riddle this though. I remember we were talking about the fourth order median filter and whatnot, but if you look at your time series right now, the one you just showed in your markdown file, looking at that, I still wonder if you can't just try a straight up, higher order high pass filter with zero phase filtering so that you're not affecting the phase of the signal, but just maintaining the nice non-shifted time domain structure. Because although I appreciate the fact that the QRS complex is a high frequency component, I still think the contaminating noise is yet higher noise. And if you look at your power spectrum, there's probably a pretty big chunk of that noise that you can get rid of that is just straight up high frequency noise and considerably clean the signal right there. So I was just gonna say before you plunge into that central fourth order moment filter and doing all this system identification and estimation of this MR signal, have you tried just straight up using
Yes, I did. I did.
And what do you get?
Either I get a non-plausible signal, or I have this just a signal that follows the same morphology, but with reduced amplitude.
But do you really care, which features of the actual ECG trace do you need? Because let's say you're saying you want.
I want to extract this R peak here.
Well, why?
To derive heart rate from it.
But you don't need to do that, because if you want just a metric of heart rate and you go and you move your high frequency noise, but you still have that threshold detection of some vaguely QRS complex looking thing in your signal, you take that, you cross correlate it with itself continuously, and the shift in the cross correlation function, the peak of that, will give you your heart rate as a sliding window.
I would be very happy to sit down with you and discuss that.
Can you send me this?
The
raw data?
Yeah, for sure. We can sit down after.
And see if that works. I feel
Okay.
that would just be straight up 4 lines of code one time.
But what would be the operational criterion that you would use to verify that this is the right detection? That
Go and count peaks yourself.
Yeah, but with 10 hours of movie, in the scanner, this is probably too long, and it would be less efficient.
Sometimes a simple solution is.
Yeah, for sure. I totally agree. I'm really open.
Just
yes.
Quick comments on this, because this is important sort of canonical discussion going on here. You're talking about developing a new method, and that's not really, we haven't had a lot of projects doing that, we have a lot of data analysis projects, so maybe that thing hasn't popped out yet. But when you develop a new method, it's critical to have simple benchmarks. Simple, straight out of the box methods that you can compare with. And unless things are really hell, those simple methods may do an okay job.
Yeah, for sure.
You can always hope to improve with more complex methods, but in a lot of problems, a linear SVM in machine learning does really well. Actually, if you push classical methods and you try to really optimize them correctly, you can often reach really close of the state of the art on lots of problems where people do crazy deep learning. So, I 100% agree with Vlad that you should try to implement baseline, simple model very well. Then there's the question, how do I know if I do well or not? And there's a simple answer for that: simulation. In engineering, when you evaluate something, you simulate the thing. Because otherwise you don't know what you're doing. So what you can do is take a clean ECG signal, add some noise with different properties to it, and run your different methods on it, quantify the results, because you have the denoise signal as an input, and get a feel of how good those methods perform in different settings. And I think you learn a lot about your methods by doing that. So I'm 100% behind Vlad on this. And if you can show that those simple methods suck, it's great. But at least it's not it's not I'm going for the latest shining with like a cool name in the new package. It's like I've properly benchmarked a number of basic methods, and I can show and actually build an intuition of why they suck. Or why they're not performing.
But by developing a new method, what do you mean?
Well, you're not developing
because
a new method, sorry,
okay.
but you're selecting a method to solve a particular problem out of a large catalog. So you need to do a proper evaluation. So that's not developing the method, it's evaluating the method.
Because I it's built upon it would be built upon a paper that has published that, and
so the paper is not science. The paper is the advertisement for the science. The science is the data and the code that reproduce the figure. If you have a paper that has that, then you'll have those simulations I was talking about, and they'll be convincing. Otherwise, you need to redo it. And then you can be really confident of what you do. Like when I got initiated to signal processing, I recoded a full thing, and it wasn't necessarily as good as the library that was at the time WebLab, developed by a brilliant group of researchers. But I was working with one of the main authors of WebLab, and he looked at what I had done, and he was like, that's good, and I was actually able to reproduce closely the figures from the book. And for me, that thing became real. I believe that method because I believe that chair is here. I can rebuild it, I can sit on it, it works, so it's a thing. Just grabbing a method and running it on data and trusting the paper, I would not, build 2 years of research on top of just that trust in the paper and the implementation.
Yeah, I totally agree, and I would like to, consider this method that you're talking about as simple. I would love that, but for me, I had better insights maybe, or it was more intuitive for me looking at this method that I was talking about than the one you're discussing.
Okay.
So
we'll chat more.
Yeah, totally.
Other feedback? ECG denoising. All right, so we're going for a break. Thank you a lot. So it's 3:30, so we're basically on time. So we'll start again at 4, and we only have 4 presentations, so we should be able to finish a little bit before 5.
I don't know if it's on purpose, but I didn't see my name in the list of
because you were on the this morning, so sorry? You were on this morning when I put the schedule.
Oh, that's why. Yeah, that's why.
Yeah, I can. Is there anyone else who isn't on the list?